Kim, how are you doing? It's Johann, your doctor friend, family, Cape Town, South Africa. Back in London, we're looking at all the new interest you're showing in this huge subject of stem cell and where it all originates from, the research going ahead, and things that may interest you personally. But before we get there, there are lots of little stories that can possibly trigger your mind into the subject of 1, stress, 2, your own children, and 3, the way forward and where the sciences are moving. I'm just quickly going to the word stress. I'm going to read off a piece of paper because it's probably a lot easier than my memory serves. But the brain, we all need to function fairly functionally every day. We've got a nice definition. Master controller of the nervous, the endocrine and the immune system. All of those are applied sciences today, and they can all be influenced in the future by what is happening in the biological research with stem cell. And stem cell development is going to probably supplant any other kind of medication if one reads between the lines. The sciences are very reserved because there are unfortunately ethics involved in the subject. But you can only imagine yourself being stressed and trying to find a medication that can de-stress you, when all you need is probably to functionally manage that little subnucleus in the deeper set of the brain, where it sets off messages of more adrenaline, more stress-added factors, which causes your cortisone levels, which is your adrenocortical function from the top end of your kidney where the adrenal gland sits, going up, and your stress levels go up. It would be very interesting if the sciences can influence those stress functions with a simple cell which can be introduced into your body, manipulated scientifically by taking a certain part of the genomes or the genetic material and tweaking it so that it can function in your brain to de-stress you when you have a trigger mechanism happening. Something like bad news about the kids back in hospital. And to diverse, I understand you're having a Kimberlin is probably going to hospital in the next few days, according to what I'm told. And you've obviously had some crisis with Matt. These, of course, the adopted children you mentioned from China. And if it should happen that any of these stem cell developments can influence their future, it would be very interesting. Whether you'll be alive to see it, it's another question. But science is moving pretty fast. So I'm going to go back to the subject of this mechanism of our stresses. It's actually a kind of way we protect our bodies against damage. That's why we have stress. And what it does is impact on everything. Not just putting your blood pressure up, not just putting your pulse rate up, but it also affects other aspects of the symptoms that can arise from symptoms relating to stimuli coming from stress. Not just the cardiac side, but going up as far as possibly your immune system being influenced. We know this. Just think of a long-distance runner. After his distance running, he comes back, he's achieved his 37 Ks, 57 Ks, he's trained, everything, he's optimally fit. But his immune system's been knocked around by physically hammering at his body. Well, that's the same process. You physically hammer yourself with stress. Your immune system compromises. That runner ends up with a flu and a cold on the first 24, 36-hour period after his achievement, having run 37.54 Ks. So it's not uncommon. These factors are what we call our sympathetic neuronal system being influenced by these hormones that we produce in our bodies. And that's all controlled through the brain. But getting back to the subject of where you wish to know a little bit more about, London is probably one of the areas where, together with the European, French, and Americans, working on stem cell information, there are questions that everybody asks about stem cells. And I'll read them out to you because it's the basic sciences that others would like to know about. What are these stem cells and why should they be important? That's the first question. Then the next one we all ask is, so what's so unique about their function and their use in our human applications? And then the next question, what are embryonic stem cells? Because we know the embryo is the beginning of man, the making of the future, whether it's animal or human. And then what are the adult stem cells, the ones that you use when you're already older and we want to take them from your own body and utilize them for purposes of present application, meaning while you're alive, elderly or younger, and you've lost a limb or something's happened to you physically, and now use your own stem cells to try and regrow one of your appendages you've lost, or rejuvenate or regenerate aspects of other organ failure when you're an adult. Then there are questions. Is there a similarity between the embryo, which is when you were being made, formulated, fertilized egg, sperm, fertilized egg, and the adult stem cell? What is the relationship? Is there a difference? And then what is this word pluripotent stem cells? It's just a simple way of trying to say what is being applied in the language. What makes a pluripotent stem cell different to an embryonic cell? And then where and what other obstacles are we about to face before these scientific potentials can be realized? What is lying ahead as problems? Well, I won't go down there. I'll start with the word ethics, as that's simple enough. We've already had the cloning of animals, and we know exactly how that goes. And the question mark of whether it should be permitted in the humans. And we don't have to go further than discuss the subject of in vitro implanting of the fetus into a surrogate mother, or something to the effect that you want to have a fertilization, but done in vitro. And of course, the problems that go with that ethical area. And those ethics apply to religion, applies to law, applies to the sciences, especially in medicine. So they're tricky little areas to cover. But I'm not going to go to all the major, minor details of each aspect of each question. I'll rather just try and give you a little basic understanding, Kim, of this stem cell pathophysiology, meaning the beginning, the things that go wrong, and how it starts, and how you make it happen, and how you invent the cell to become something else. I'm going to show a little simple diagram. You can just have a look at it. I've written it with a black chalky pen. It's not exactly perfect, but about there you should be able to read it. I'm not going to hold it up forever and talk behind the screen, but it's a basic explanation. And I'm going to put it down. I'll put it up again in a few seconds. This is where life begins. The sperm enters the ova. The female is fertilized by the male. Nature. The fertilization takes place. And about 4 or 5 days later, there's a cell mass that's created. And we call that the blastocyst. The blastocyst is actually about to be implanted into the uterus, where it then grows and it develops embryonically, as we would expect a human to go through its various phases of embryo development. And of course, after 9 months, we have what we call the species, human, maybe animals, in different years, months. The blastocyst, getting back to that. The blastocyst, as on this picture, and I'll show you again, is on the 5th day. And that 5th day is where we would take the cells that are in there developing and multiplying, they're differentiating. And scientifically we've used this particular aspect to try and take cells out from there and then utilize that mass to actually grow more cells. Cells of specific needs, like if you want a cell to grow the brain, want a cell to grow the bone marrow and blood cells. And the bone marrow blood cells are a whole area of what we call, and it's a very clear place for you to realize that our differentiation of that particular blastocyst changes, and it becomes a very interesting choice of various tissues that change from that blastocyst while the implanting of that blastocyst happens in the actual uterus. We develop, it changes into all kinds of ectodermal cells, mesodermal cells, and endodermal cells. Now this sounds like scientific hogwash, but it actually basically is. Neurons, which affects the neuronal system, the brains, the spinal cord, and nerves, which is the ectoderm. And then the mesoderm forms the blood cells, meaning all the bone marrow and peripheral blood circulation cells we find. And then there's the endoderm, which is the organ side, and especially the liver gets covered on that aspect. Now by the time this development of this fifth day blastocyst has happened, there are already in place cells that can be determining ectoderm, neuro, brain, mesoderm, bloods, endoderm, liver cells. So if we are able to extract some of those cells out of that blasted cyst, then we can take them out and we can develop those stem cells into specialized or unspecialized cells in the laboratory. But knowing that the blastocyst already has made its choices, we can't change it to something that we want to make it become as we please. So we've got to engineer it and manipulate it. But normally, these cells that are in that blastocyst, those little, call them early development of our human cells, they differentiate and they divide. But they do this almost continuously. But they can also not be active. They can become an inactive aspect. And if those inactive cells remain inactive, changes happen, which we begin to start recognizing a little too late or too early. And we talk about defects, in vitro defects. Now these are things that we can pick up in our modern science by amniocentesis, by direct cell, by checking on the actual cells in the blastocyst. We can even make predictions. Is a possibility going to be that one of these cells will change into something which isn't quite the pattern expected from the genetics in those cells? Well, that's where we are moving into the future. And that is probably where we're going to use the word reprogramming. And that reprogramming of specialized human cells is what we call induced pluripotential stem cells. That is where the legal, the ethics, and everything holds. Because those supposed reprogrammed cells are going to be further advanced than 2006. Why I mentioned 2006? Because that was where it all began in terms of the new science of stem cells. If we go back to 1981, that's when the mice embryo stem cells were used for all kinds of manipulation of the genotyping and the DNA, and all affected the coloring, the sizes, the multiplication, 2 heads, 5 tails, anything like that went. But that was 1981. That took us further down the science trip to 1999. And that's where we started the huge ethical problem of human embryonic cells taken from that area of blastocysts, 5th day, and reproducing, taking those little blastocyst cells out of the blastocoel. And I repeat, I'll show you the picture again, the blastocoel. If you look at the picture in here, those are the little modular cells, and they all contain DNA, which has all kinds of typing on it, all proteins in the body required, all nature of our color, eyes, everything else is all printed into that particular DNA. It all lies in those little cells within this blastocyst. There's a wall around it, if you can see there, and it clearly will show you the tropofectron derm, which means the skin on the outside of the cyst. All that happens is these little cells, or bodies, or stem cells, are actually with a pipette almost. And of course this is micro measurements, sucked out and introduced into a growth media. They use like a petri dish. They can use blood, they can use all kinds of things, and they regrow those under controlled circumstances, which is where we are in 2006, which is when those cells are reprogrammed. And that specialized human cells we are going to reprogram are the ones we can say, if the mother chooses, father chooses, before the fertilization, would you like a blonde child? Would you like a blue-eyed child? All these kinds of developments are in the making. They're probably, as we speak, quite advanced. Now, how does this actually apply to you in your circumstances and your family, Kim? I suspect it could be useful in terms of, maybe you won't live to see it, but there's a chance, if these new pluripotent stem cells are utilized from anywhere to reinsert and reorganize tissue in our human bodies, like cancer cells that are multiplying, and taking the cancer cells that are there and looking at the genotypes, looking at the DNA of what's turning them wrong and what's making them go faster and what's multiplying them, and then introducing a stem cell that can take over that function. And that stem cell can then suppress the activity of this multiplication factor. In other words, the cancer won't grow. Or it can give a message, say, kill off the remaining cancer cells, let them die. All this is a potential of the future, and it's happening as we speak, I think. So I think in your particular instance, where you've got children with certain defects, heart, cardiac, it could happen one day that they could choose, and they could choose somewhere along the line that defects that may exist can be re-manipulated or changed or removed, organ removal, re-stimulating that organ, regrowing that organ. It's all very much a potential for the future. So I don't want to dog you down and make you feel that all these huge applications are something that's too far gone and too far-fetched, but I can speak of something that's happening as we are on this particular program from London. Applications using stem cells to re-epithelialize skin is happening as we're speaking. In other words, patients with massive body surface burns, where no potential chance of doing transplants or grafting or anything else is going to be of any use. It will be not considered as life-saving, but if it's correctly applied in the right moment and time of the injury or burn or other, the new skin will be able to replicate the original state of the skin of the person before he got burnt. It could be copying almost what was his previous skin condition. It may not look like it for the first 9 months, a year, but eventually the pigmentation will pick up from the genes that are recognizing other cell functions, or the new cell picks up the other cell functions and causes a change. Like, I'm brown skinned, I'm lighter skinned, I'm darker skinned. I don't say this is a new way to create a new race, but it's not impossible. We could create a new Mars people and they could turn out to be green. So it could be very interesting. I wouldn't like to say what else we can invent with it, but that is one of the recent and possibly nearest applications we've got. But I wish that one day, if you can live long enough, you can have this applied for the use of your own children. And hopefully they will also benefit for anything from a limb, to defect brain, to vision, teeth, limbs. So stem cell development has reached a stage of not questioning anymore, it's having to control it. And that control aspect is going to be an unfortunate message for people of all kinds of background, from religion to politics, to probably ethnobotanists who work in a laboratory, if not botanists working with plant life. As we know that plant life is already having problems from the genotyping and the particular kind of new DNA GA foods, because they are actually trying to create foods and artificial, call them biological products, which could produce things that they weren't made to produce in the first instance. So a rose could smell differently because you're going to create a new smell for the rose. That's been happening for years. But this is done almost instantaneously on the stem root, using DNA and using all these kind of stem cells. That's biological plant life. But the marriage of all these things are happening so quickly that the days gone by where we were cloning sheep, mice, 2 heads, 6 feet, it's now going to be humans. So I think in the religious sense of the word, which may be something for you to chew on in terms of checking out what's going out there in terms of the view of the church and religion and the view of science in this manipulation in the future of stem cells and the human race. Hope you enjoyed that. Have fun. Thanks, Kim.